Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Akoya Biosciences webinar titled Reprogramming the Brain Tumor Microenvironment to Enhance Anti-Tumor Immunity. This is Liam Sanyo from the events team here at Scientist.com, and I'll be the host today. We're fortunate to be joined by Dr. Theo Mantamediotis, who's Associate Professor and Laboratory Head at the Doherty Institute at the University of Melbourne, who will show how recent discoveries about the brain tumor microenvironment can drive new therapeutic strategies to overcome tumor immunosuppression. And with that, I'm very pleased to welcome Dr. Theo Mantamediotis. Theo, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, thank you, everyone. So I will tell you about some of the work we've done using the Akoya multiplex immunohistochemistry platform. The topic is reprogramming the brain tumor microenvironment to enhance anti-tumor immunity. And I'll just give a little bit of an introduction to my laboratory. So we're focused on brain cancer mainly. We use a lot of tissue analysis, which is really a lot of the focus on the topic today. We also use organoids derived from brain tumors and also neural stem cells to understand parallel mechanisms operating in cancer and in brain tumor-derived stem cells. Also, we use mouse models, and I'll show you a bit of the data we have using, again, the Akoya multiplex IF or IHC platform. So what I'll do is break up the talk into some subtopics, of course, focusing on our research and technical use of the multiplex immunohistochemistry. But I wanted to give you a bit of background on brain cancer, the sort of previous work that's been done, and the understanding of brain cancer biology using omics, genomics, transcriptomics, and so forth, and how then that relates to the way that a complex heterogeneous microenvironment in the tumor operates. I'll tell you a little bit about the tissue analysis we've done, the spatial proteomics, how we've used it to cell map, but also to investigate cell function in the tumor microenvironment, and focus a little bit on mechanisms that we've paralleled multiple experimental models, including the multiplex immunohistochemistry spatial biology, to understand the role of the macrophages and the mechanisms driving immunosuppression in the brain tumor microenvironment, with a little bit of cell signaling mechanisms that drive some of these macrophage functions. And I'll end the research part of my talk really talking also about a kind of parallel interest in enhancing the anti-tumor immunity relates not just to immunosuppression, but also in looking at how immune cells traffic macrophages mainly, but also relates to lymphocytes or T cells. So brain cancers, there are many different types of brain cancers. In fact, and you can see on the left panel here that this is so-called primary brain cancer, which is really a cancer that begins and typically and rarely metastasizes out of the brain. So it's confined to the brain, and there are many different names that are used. They're known as gliomas, which refer to both low and high grade cancers. They're also called astrocytomas, and they're the most common type. As you can see around here, there are also various cell types that are affected in terms of oncogenesis, which create tumors which are much more rare. The most common and aggressive form that develops either from a low-grade glioma or astrocytoma is glioblastoma, and a lot of my talk will be focused on this particular disease. There are also, of course, metastatic brain cancers, and we've studied some of those. I won't tell you much about those today, but they're, in fact, in terms of incidence, of course, much more common than primary brain cancers because they relate to the primary cancers which are typically the most common, such as lung, breast, and melanoma. And at late stages in a subset of patients, there will be metastasis to various organs around the body, including the brain. Now, in terms of glioblastoma or GBM, what's quite sad is that it's a very difficult disease to treat. And in fact, treatment has not changed since 2005 in a substantial way, apart from clinical trials, which in some cases have shown such some promise, but the standard therapy really has not changed worldwide. And standard therapy involves maximal safe surgery as quickly as possible after diagnosis, which is usually by MRI or some sort of imaging protocol, and then followed by radiotherapy and temozolomide, a chemotherapeutic DNA alkylating drug for a certain time, and then maintenance of some treatment perhaps, but also monitoring of patients. But unfortunately, recurrence or relapse typically occurs within 12 months. Sorry, I have a spelling mistake there, moths. And you can see here the representation of the so-called advanced protocol since 2005. There is some improvement in therapy, and this is a survival Kaplan-Meier curve showing you a large population of patients that this treatment was better than the previous treatment before 2005, which really focused mainly on radiotherapy and some use of chemotherapy, but not under this so-called STIP protocol. In terms of pathology of glioblastoma or astrocytoma, you can see here there's normal brain, and this is a hematoxylin and niacin stained section. The pathologist would look at if there was a normal tissue anywhere in a specimen or a biopsy, and then there are various stages of disease progression, which a pathologist using standard immunostaining, H&E staining can see changes in the cell morphology, increase in cell density, evidence of proliferation, and so on. And over the years, there's been, I guess, better appreciation of the sorts of morphological features that can be recognized and help with diagnosis. With the advent of molecular tests and understanding of the underlying genetics of glioblastoma, there's been various mutations such as P53 mutations, IDH mutations, and so on, as you can see here, which are some of which are now entered as a standard pathological test to aid a standard microscopic diagnosis. And in terms of omics and genomics and transcriptomics, in the last 15 years or so, there's been a huge number of samples that have been glioblastoma samples that have been analyzed at DNA and RNA level. And what you can see on the left here is the columns, each individual column represents a different patient tumor, where they've done DNA sequencing and identified mutations, deletions, or amplifications of various genes. And they've been clustered with the gene names shown on the left here into different modules which relate to cell signaling or cell function, such as receptor tyrosine kinase mutations or PI3 kinase, MAP kinase, and so on. So there's a better understanding of the different subsets of genomic differences in glioblastoma tumors. An advance similarly with RNA sequencing allowed researchers at least and clinicians to understand that there are four major glioblastoma subtypes, which you can see on the top here, based on the expression of many different genes, but some of the major genes shown on the right-hand side here. So that's allowed us to understand the biology of GBM, and in fact there are four subtypes. Unfortunately, that hasn't led to real clinical application of this knowledge to this point at least. And I guess the omics has been very powerful, as we've seen in many different diseases, especially cancers, including glioblastoma. The issue with genomics and RNA sequencing has been that it's been done on so-called bulk tissue as a bulk tissue analysis, and really that represented here, where you have a tissue with your cells or different components of the tissue sitting in some sort of order, functional order. And really omics has relied on blending the tissue, in fact, and then extracting the RNA or DNA and sequencing, which is very powerful. It allows high throughput deep analysis, of course, of these macromolecules, but the disadvantages are, of course, the spatial context is lost. Heterogeneity of the tumor, in essence, apart from the molecular heterogeneity, is lost. And certainly functional aspects such as cell-to-cell interaction or cell to extracellular matrix interaction is also lost. And I really like this statement from a review written by Jorge Nievo and James Hicks: when it comes to genomic analysis of tumors, don't buy bulk. And I think that's a lesson that, of course, spatial biology is filling all those gaps in terms of understanding the complexity of glioblastoma. And really the way we have investigated glioblastoma biology is we got on very early to the opal immunohistochemistry, which Akoya now runs across their platforms. And the way it works is you can do whole slide staining, and you can use FFPE tissue. So we used archival tissue in many cases, often 10 to 15 years old from our anatomical pathology lab. You can use automated stainers, automated image acquisition, and you can do digital image analysis using various softwares, some commercial, some open source. And really what we and many others have realized also is that the power of spatial biology analysis or spatial tissue analysis comes from overlaying other sorts of analyses on the tissue, perhaps also in vitro analysis to sort of couple over the top of the spatial analysis to understand very specific functions at a single cell level, ultimately aided by the spatial biology. And this is an example of the Akoya 6-plex, which is actually a 7-color because it includes 6 antibodies plus DAPI. And this is an example from my laboratory showing a glioblastoma tissue where we've used 6 antibodies here and looking at various cell functions, whether it be immune cell infiltration. You can see the detail and the heterogeneity is incredible. And of course, coupled with detailed image capture and digitization of the image and software analysis, you have a very powerful approach to trying to predict functional interactions occurring in this very complex tissue. And very simple aspects, of course, that many people do is enumerate or count the number of immune cells infiltrating into the tumor. And that's, of course, important to understand in the context perhaps of disease progression, but also in the context of immunotherapy. And in this example, I show you one of our studies where we looked at many different individual tumor samples. So it's high throughput using tissue microarrays, where we looked at microglia, the intrinsic myeloid cells in the brain, macrophages, which are coming from the circulation, and T cells. And of course, what you can do is count every single cell in every single sample. And what we see, unsurprisingly, and this is known in many different cancer types, that during disease progression, where you have non-tumor tissue adjacent to the tumors that we were able to analyze, and then the different grades or stages of the disease, all the way to grade 4 in the case of glioma, which is in fact glioblastoma, which is grade 4, there's an increase cell density of the different immune cell types. And what we also notice, and many other people have reported this also, is that macrophages are by far the dominant immune cells that infiltrate into the brain tumors. And you can see that by comparing the scale of the y-axis from the macrophages to the T cells in terms of cell density. But what we wanted to do was not just count cells and try and look at disease progression. We were really interested in predicting cell function or determining cell function in that very complex microenvironment. And the way we decided to do that is not just use, for example, cell proliferation markers such as Ki67 or PCNA. We were looking at the cancer hallmarks and looking at what sort of other functions can we look at. And we were interested in cell signaling, since cell signaling pathways dictate gene expression changes and therefore functional changes. And one of the pathways we are interested in is, in fact, 3 different cytoplasmic signaling pathways, which you can see here, the cyclic AMP, the MAP kinase, and the PI3 kinase, in fact, which all converge up onto this transcriptional signaling hub via the cyclic AMP response element binding protein, or CREB. And that's been an interest of ours for many years. And this is a really great transcription factor to look at. In fact, it's essentially ubiquitous, major functions in the brain because it's phosphorylated, and they're really great antibodies that can distinguish the phosphorylated form compared to the non-phosphorylated form. And phosphorylation means that the transcription factor becomes active, transactivates many different target genes. This is an example of the sorts of studies we've used to look at CREB function. We've looked at the brain function for many years, either cell survival within the neurons, looking at T cell function, many in mouse models, but also we've become interested in cancer via our interest in Krebs' role in neural development, neural stem cell biology, various models again in zebrafish and mice. And that sort of led us on to the idea that maybe we should look at Krebs in cancer, since we noticed that lots of phospho-Krebs expression occurs in higher grade tumors in glioblastoma compared to lower grade gliomas. And some of our first work was looking at in vitro experiments, looking at glioblastoma cells. And what we saw is that Krebs regulates cyclin D1 mainly, and also co-regulates cyclin B1. Seems to be very important for regulating cell proliferation in glioblastoma. And then over the years, we developed various mouse models, including one which was PI3 kinase driven, in which we were able to genetically delete Krebs. And what we showed is that the mice with aggressive tumors, when Krebs was deleted specifically within the cancer cells, the mice showed extended symptom-free survival. So Krebs is really an important factor in driving cell proliferation in brain tumors, but not just brain tumors, but in fact other tumors, as some others have shown. But that was our main interest. And I guess over time, we also became interested in understanding how this could be applied in the context perhaps of a clinical benefit, apart from targeting Krebs for stopping the tumor cells from growing. We knew already that there were lots of studies showing that Krebs is really important transcription factor in regulating immune cells. So we then wanted to go on and look at whole tissue, to look at not just cancer cells, but the immune cells. We started off by using immunohistochemistry to look at cell signaling. Of course, that's a single color, chromogenic assay. And what you can see here is an H&E stain of glioblastoma tissue, and then using phospho-specific antibodies to look at the either the MAP kinase pathway using this antibody, the PI3 kinase pathway, and also Krebs, and a cell proliferation biomarker, PCNA. And ultimately, we were interested in immune cells, but what we saw in the tissue, which was quite remarkable, is that there's already heterogeneity at the level of cell signaling across the tissue, often mutually exclusive, sometimes overlapping between the major cell signaling pathways shown here. We went from monochrome immunohistochemistry to applying the Akoya 7-color opal system to look at glioblastoma tissue. And this is a large piece of glioma tissue, about 1.5 centimeters in length across this direction. And what we used, again, is these phospho-specific antibodies, and also a biomarker for endothelial cells, which are the blood vessels, which you can clearly see in cross-section in many different regions of the tumor. And what's remarkable, again, is the level of heterogeneity that you see across the cell signaling. So even though in this region here, for example, there are essentially thousands and thousands of glioblastoma tumor cells, what you see is that almost every single cell perhaps has a different cell signaling pathway that's activated at that moment in time that this tissue was captured. And it seems to correlate with specific regional pathological hallmarks. And this example shows you that same tissue where we've stained with Masson's trichrome staining, showing you collagen in green, and the cellular, highly cellular regions in the eosin stain here in red. And then the multiplex, which I showed you in the previous slide, showing you the cell activation pathways and the blood vessels. And what we were able to do using software is map every single cell and look at which pathways were activated or co-activated. And what we were able to do is essentially predict function of every single region here that seemed to correlate with specific histopathological hallmarks. For example, tumor cell dense regions show activation of all or both major cytoplasmic pathways and Krebs. And this really relates to high cell proliferative activity and high metabolic activity. Where there was lots of extracellular matrix and angiogenesis, so in these darker regions here, what we saw is not much PI3 kinase signaling, but lots of MAP kinase and Krebs signaling, and that seems to correlate with proliferation, but more specifically with angiogenesis. And then there was a very interesting interface between the extracellular matrix rich region, and the tumor cell rich region, where there was a different signaling pattern shown in lime green here, and that seemed to indicate low proliferation, probably differentiation and migration, but certainly some sort of communication between the extracellular matrix and the tumor cells, which was programming the cells very differently to other regions. And we're able to test that by using organoids or neurospheres. And what you see here is just the two major pathways, MAP kinase and PI3 kinase in different colors of an organoid. And this is from glioblastoma cells, showing that there is a relatively uniform activation of the pathways with some differences around the edge exposed to the growth factors. But when you put these organoids into a collagen matrix, semi-solid matrix, what you see is that the cells on the edge begin to migrate, and they show very different cell signaling activation program compared to the cells which are not migrating. So it kind of at least recapitulates some of the aspects of the tissue that we saw and the cell signaling profiles which are activated. And I'm going to jump a little bit back to the immune cells because it relates both to cell signaling and then how we use that to understand immune cell function. Is that what we noticed when we were doing our sixplex opal is that macrophages and T cells showed phosphocreb staining in different regions where either macrophages or T cells were present. But what was really remarkable when we looked across the whole tissue again at cell density of phosphocreb positive cells within either the macrophage subset or the T cell subset, and I'm sorry, you can't see the labels here, is that we saw an increase in the proportion of phosphocreb positive cells during disease progression. So it's not only that the immune cell numbers increase across disease progression, also the activation as measured by phosphocreb increases. So the immune cells actually become more active. What that specifically means in the context of phosphocreb, we're still exploring that aspect. One thing we did want to focus on more was the macrophages, given that they are by far the dominant immune cells infiltrating into glioblastoma. We developed another panel, sixplex 7-color panel, to look at whether macrophages were pro-inflammatory or anti-inflammatory, and there were also the mixed phenotype using the biomarkers shown here. And you can see an example here of glioblastoma tissue, and you see again, the heterogeneity of the macrophages. So this is only looking at CD68 positive macrophages and the various states at which these macrophages are in different regions of the tissue. And what we saw is that during disease progression, there was a shift from in low-grade tumors were dominated by the pro-inflammatory macrophages. But when you went to the GBM, the grade 4, the most advanced stage of the disease, the macrophages were in fact shifting towards an immunosuppressive CD163 phenotype. So clearly there was a change in the macrophages during disease progression. When we looked at phosphocreb in the macrophages, to go back to look at what phosphocreb potentially is doing in these macrophages, or Creb is doing, in fact, we saw that almost all macrophages in glioblastoma were phosphocreb positive. So these are all phosphocreb positive, and then looking at the subsets of macrophage status, and nearly all of them are the anti-inflammatory or immunosuppressive subtype. So we were thinking, well, Creb may be driving that immunosuppressive phenotype in macrophages. So what we wanted to do was to couple that with in vitro experiments where we were wanting to look at the interaction and potential change in function of macrophages based on their interaction with glioblastoma cells, and using various experimental platforms to study this, including, of course, the spatial tissue analysis, but we were also using various other platforms. And in this example, I show you how we've kind of scaffolded different experimental platforms to understand a very complex question. And I won't show you all the data, it's unpublished at this time, but what we did was grow patient-derived glioblastoma cells. We tested a number of different GBM cells to see how they would interact with the macrophages, and we take the conditioned medium from these, put them onto macrophages, and we used both THP1 cells, but also patient or healthy individual-derived peripheral blood mononuclear cells. So we were essentially generating tumor-educated or tumor-associated macrophages in this context and taking the macrophages for this omics analysis. And we've identified a series of genes that seem to be responsive to the glioblastoma cells within, so macrophage-specific genes, and also genes which seem to be regulated by Krebs. So we're trying to understand that whole kind of regulatory mechanism of genes that drive immunosuppression in macrophages in response to the tumor. What we're also trying to do is go back to cell signaling and understand within these macrophages, which are responsive to something that's stimulating them from the GBM cells, what exactly is going on. And what we saw is that most of the macrophages which were phospho-Krebs positive were in fact negative for MAP kinase and PI3 kinase, which was a little bit surprising. So then we went, well, let's measure or use drugs, in fact, inhibitors of each pathway to test which pathway possibly is operating in macrophages that is also driving Krebs transactivation and gene expression. And what we saw is that when we use a cyclic AMP inhibitor drug, H89, we could block or at least lower the level of phospho-Krebs expression and not with the other pathway inhibitors. So it seems that cyclic AMP is the cell signaling cytoplasmic pathway that activates Krebs, which then goes on to activate various target genes within the macrophages that regulate immunosuppression. And what we've discovered is there's a whole lot of cytokines and chemokines that Krebs regulates specifically in the macrophages, which we're now still exploring. And I just wanted to move then on a little bit about towards the second part of my talk, which is really about we know that macrophages drive immunosuppression, which is bad. Can we reverse that? Possibly by targeting Krebs. So that's one aspect we've now discovered. But also what we realized is when we were looking at the tissue, well, we need to have T cells or lymphocytes that are effective anti-tumor killing cells. But for them to be killing a tumor cell, T cells need to be interacting physically with the cancer cells. So where are they was really the question. Where are these CD8 positive cytotoxic T cells within the tumor? And what we noticed across very early experiments when we would look at glioblastoma tissue, again, this is using the OPAL system here and showing you a few markers only, which are all T cell specific, CD3, CD4, and CD8. What we noticed is that most of the T cells were in fact seem to be trapped around blood vessels. So these are blood vessels, capillaries, angiogenic regions within glioblastoma. And you can see the purple and green. And when we looked at other staining and looked more closely around blood vessels, what we'd see is lots of collagen using Masson's trichrome stain, lots of T cells shown in white here with the CD3 antibody. Also a fair few macrophages using CD68 as the biomarker. And we decided then to take that real spatial analysis by doing infiltration analysis, where we'd mark the blood vessels as 0.0 or ground zero, and you can see a few capillaries around here shown in orange. And then we generated 25 micron bands around that would extend out to about 300 microns in most cases across different areas of the GBM tissue we were analyzing and across many tissues. And we'd look at the infiltration density, if you like, cell density of either T cells or macrophages, where on the x-axis, the 0 or below 0 is either within or around the blood vessel or capillary. And then as you extend to the right here on the x-axis, you're moving away from the blood vessels. And what we saw is that T cell density drops off enormously as soon as you get about 25 to 50 microns away from blood vessels. Macrophages, on the other hand, seem to be much better at penetrating and migrating from the blood vessels into the tumor. So that was an interesting finding. What we then did was we went back to our macrophage-specific OPAL panel, where we could distinguish pro-inflammatory or immunosuppressive macrophages. And what we did, again, the same infiltration type of analysis. And what we saw was that pro-inflammatory macrophages seemed to be pretty much around the blood vessels or in the blood vessels. And that for us, we're interpreting that at the moment. That means that the monocytes or macrophages arrive as in a pro-inflammatory state. But as they begin to move outside of the blood vessels into the tumor, they shift their phenotype. And we can see here, if you're looking for the immunosuppressive macrophages, what you see is they're present in higher numbers away from the blood vessels to the right-hand side within this box here, which means the macrophages arrive in a pro-inflammatory state, but as they migrate into the GBM tissue, they switch to an immunosuppressive state. And immunosuppression is bad if you're thinking about your immune system or your cytotoxic T cells killing tumor cells, or perhaps immunotherapy that you want to sustain a high level of immune activity against the tumor cells. Unfortunately, this immunosuppression seems to be present across the tumor and probably dictated by the macrophages. So that's essentially using the 6 antibody 7-color oval platform. We were lucky enough to have contact with various people at Akoya Biosciences, both here in Australia and in the US. And we were able to access and help develop various neuro-oncology panels, both for mouse and human studies. And this is the new generation PhenoCycler Fusion system from Akoya, which allows you to do anywhere between 15, which we've done, up to 50, and I know others have done up to 100 antibodies on a single tissue. So it's a very powerful way of looking at very complex tissue with multiple biomarkers. And the workflow is shown here, where again, it's very similar in the early stages where you have your tissue on a slide, and then you put in an antibody panel. In this case, the antibodies are barcoded essentially, and there's various cycles which stain using 3 antibodies at a time, which is all automated in the Akoya system. And then your output is a multicolored image, which allows you to look at every single biomarker in that system. And so far, we've managed to do some analysis using mouse brain tissue, and the panel shown here is I think it was a 43 antibody panel, and what you see on the bottom here is mouse coronal sections of mouse brain with different tumors. Sorry, you see the tumor in the purple or pink here in different models. So we're comparing different mouse models of GBM with GBM tumors, as well as a normal brain here on the bottom right. And what you see is that you get spectacular images, but the real power is in the depth of analysis, since every single biomarker can be analyzed at single cell level, and you can look at proportions of different biomarkers or cell types depending on the software and the sort of analysis you want to do. And you can see some of those cell types are shown here. You can create UMAP or t-SNE plots to give you a visualization of the complexity of the tumor tissue or the cellularity of the tumor tissue. And you can map every single biomarker to give you a comparison of where different cell types or biomarkers are in relation to other biomarkers in the tissue. What's very powerful about using mouse models is that you have the whole tumor essentially as a section, and importantly, you also have the periphery of the tumor, which interacts with the non-tumor part of the brain. So we can look at the periphery of the tissue and interaction with the healthy brain tissue as well, which we're beginning to do. We've also used the PhenoCycler Fusion, where there was a 66 marker or biomarker panel developed. We also had a subset of 57 biomarkers that we used, and you can see that each antibody or biomarker is shown here into its functional sort of subset or module, where you can have immune cell profiling, lymphocyte specific or immune activity, structural, metabolism, neuronal specific, and also myeloid or macrophage specific as well. And this is an example of, actually this is from one slide where there were 2 sections or 2 fragments of a patient glioblastoma tumor. And you can see it's a huge area of tissue, almost 2 centimeters down this way and almost 1.5 centimeters across. And we were able to map every single cell across that very large region of the slide. And just to give you an idea of what we can see using this PhenoCycler Fusion multiplex panel. This is an example of some very interesting regions we're focusing on. As I told you, the perivascular region, which seems to trap lymphocytes and has a lot of collagen around it. In this example, you see the individual biomarkers, which also relate to the image on the right, where we can see the CD4 T cells, the CD8 T cells, B cells in this case, macrophages, and the purple around here is GFAP, which is a biomarker which is typical and very highly expressed in glioblastoma. And you can see the sort of detail across here. So we're currently enumerating cell number, cell type, and so on, and measuring distances, once again, using this PhenoCycler fusion derived data. So I'm nearing the end of my talk. What I wanted to sort of mention to you before I finish up is that I think the power in using spatial biology for us has been in understanding function, not just counting cells, but really trying to understand functional interaction. And I should also say we're beginning to use antibodies against soluble factors such as cytokines, and we've successfully managed to do that for interleukin-10, so we can see where interleukin-10, an anti-inflammatory cytokine, is produced, and zero in on the cells even that are probably producing it, given we can see concentration gradients of interleukin-10. And the power really comes when you really relate spatial analysis and use it in the context of publicly available genomic data, or perhaps your own genomic data, single cell sequencing data, and of course cell biology, again, which is really important for us to look at function, especially in the context of the cell signaling pathway I mentioned earlier. Also in the context of using it, spatial biology in organoids. It's a more simple system that one can control, put the organoids into a context which you can then measure the output in terms of cell signaling or cell type distribution in the organoid. And we're now beginning to couple that with three-dimensional clearing tissue, mouse brain tissue as shown here. And ultimately, we'd hope to perhaps even consider three-dimensional analysis of spatial cellularity and perhaps even cell signaling. And ultimately for us, given I mentioned cell signaling and CREB being a transcription factor, which really important regulator of both cancer cells, but also the macrophages, is that we're ultimately going to try and couple that with spatial transcriptomics. So that would be incredibly powerful, given that we're also have a cell signaling phospho-specific antibody antibody panel. It'll be fascinating to see if we can relate specific cell signaling pathways or transcriptional activity to specific gene expression at a single cell level. So in summary, what I've tried to tell you is that we've used spatial biology in the context of the Akoya platform to define properties which we couldn't define otherwise, certainly in a high throughput, high dimensional way. But coupled that to understand things we were already interested in, that is cell signaling and transcription hubs, which we've now realized are really important in the context not just of the cancer cells, but also macrophages. So can we target CREB to regulate or reverse immunosuppression for that matter? And also the spatial part would have been almost impossible to do in a detailed way using other technologies, is that the extracellular matrix, especially around the perivascular regions, seems to be some sort of barrier or sequesters some sort of factors that regulate immune cell infiltration. So clearly, and many others are now thinking of that in many different contexts of different cancer types, that you need to also think about immune cell trafficking. So we've got a potential target that we've realized exists within glioblastoma. And I just finish up by thanking a few people, and I'd like to do that. Samuel Widodo, PhD student who's just finishing, Maria Dinevska, both who have really driven all the research I've shown you here. I run a very small laboratory. These PhD students have been incredible in really driving a lot of the development of the spatial biology capacity we have and how we use that. The immunologists in my department, Laura Cook especially, but also some of the bioinformaticians, Dominic, who helped us with analysis, as well as some of the other immunologists. Special thanks to Marlene and Lincoln, who we've collaborated with. I didn't mention Stan Stiley, but he's been a long-time collaborator. Anyone that looks at our published work will see that we've published 20 papers in the last few years with Stan and kind of driving that whole spatial biology analysis. And various other people at the Peter MacCallum Cancer Centre, immunologists and cancer specialists, the Histology Centre, which really runs the platform that we use in terms of the Akoya system, and also Akoya Biosciences for giving us the opportunity to work on the phenocycler fusion system using those mouse experiments that I showed you towards the end, and some of that recent human GBM analysis. Jackie Maurer in Australia, and also Dimitro Niati, Shekhar, and Oliver. So I'll leave it there. Thank you for listening.
Well, thank you, Theo, for a really great presentation. Some really interesting work there. All right, so we'll start things off with an interesting question here. Theo, what were the most critical factors you considered in developing a multiplex immunofluorescence panel for glioblastomas?
Yeah, so we started off simple by looking at the immune cells, macrophages and T cells, but then really in trying to understand the mechanisms that underpin glioblastoma biology, given that complexity and heterogeneity, we decided to take a modular sort of view of developing our panels. So we had our immune cell panels, then we developed our myeloid-specific panel, the cell signaling panel. So really it's about distinguishing, many different levels where we can mix and match some of the antibodies across multiple opal panels in this case, to really understand deep biology. And that's been the real strength and what drives us, and really what we consider is understanding function ultimately.
All right, great answer. Another good one here. So this person says, recent work has shown that PERK as a potential predictive biomarker for GBM with the spatial proximity between PERK expressing tumor cells and infiltrating myeloid cells and microglia impacting response to immune checkpoint therapies. Can you comment on TAM polarization and tumor cell PERK status as a predictor of checkpoint therapy outcomes?
Yeah, so that's very close to what we've done, as I showed you, is looking at cell signaling. We haven't looked at that specifically, but that others have, and actually an amazing study published in the last few years by Fabio Iwamoto and Raul Rabadan and Adam Sonnabend demonstrated that neoplastic cells, as you mentioned, the cancer cells in recurrent glioblastoma, so that's when it comes back after therapy, the relapsed tumor, exhibit that high level of phospho-ERK expression, which is really MAP kinase signaling, of course, localized to the tumor-associated macrophages. So these macrophages that are close to the high ERK-expressing tumor cells seem to have elevated MH class 2 expression. And this is important because those patients who showed that particular phenomenon were the patients that responded well to anti-PD-1 immunotherapy, for example. So it's a really important aspect to look at cell signaling, and clearly, that is going to become more interesting as immunotherapy becomes more important in certainly brain cancer therapy and clinical trials, of course, because we want to know in the clinical trials what's going on in responding versus non-responding patients.
Yeah, definitely good points. All right, next one here. Are there any other potential biomarkers that could be used to predict response to immunotherapy in GBM?
Yeah, I'm not entirely aware what others are doing, but I think I mentioned briefly that we've now developed a panel where we look at soluble cytokines produced in the tumor by presumably the macrophages in many cases, perhaps some of them by the tumor cells or other immune cell types. So the detection of soluble biomarkers, I think, are great, and we've shown, at least for interleukin-10, we can do that. DNA damage is another one. So H2AX is actually in the phenocycler fusion panel, given that if you're treating the tumor or the the cells, immune cells are potentially killing tumor cells. You could measure H2AX levels, which should go up if there's DNA damage in the cell. T cell activation biomarkers would be fascinating to use in the context of spatial biology. I mean, T cell activation biomarkers are standardly used by immunologists using flow cytometry. Of course, that's a single cell ex vivo in vitro type of experiment, but to be able to look at T cell activation in the context of tissue would be fascinating in patient tissue or treated patients.
Yeah, definitely. All right, and I think we'll close things out with a sort of forward-looking question. So how do you see the role of spatial biology and single-cell omics evolving in future glioblastoma research, and especially in relation to deciphering the interplay between tumor-immune ECM interactions?
Yeah, we still don't know although we're predicting based on our studies so far what the extracellular matrix potentially is doing. Clearly, it's changing the cell signaling profile of the tumor cells that are interacting with the ECM. But of course, what we also, and I didn't really show you much of that, what we also know is that the ECM-rich regions, so it's a fibrotic chronic inflammatory response that occurs in glioblastoma, these regions, they seem to expand over time, we presume, and they seem to be a reservoir for immune cells as well. So there's something going on. And also, I guess, CD44, which is highly expressed in cancer cells, the major ligand for CD44, which is expressed in the glioblastoma cells, is hyaluronic acid, which is a major ECM protein. So what actually is that signaling to the cancer cells? We don't know, but that's the sort of fascination in terms of not just immunity, but also specifically cancer cell function that changes in response to the extracellular matrix interaction.
Awesome. Yeah, lots of really interesting work yet to be done. But thanks so much, Theo, for your time today and for the great presentation. It's really been a pleasure having you with us.
Thank you.
A big thanks also to the audience here for participating. So in closing, we hope you enjoyed this Scientist.com and Akoya Biosciences webinar, and we'll see you again next time. Have a great day, everyone.